nucleic acid, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. DNA, it is act as genetic material that transfer from parents to offspring. RNA, it also act as genetic material. Besides, various functions are there, like messenger molecule mRNA, adapter molecule tRNA, messenger RNA and the transfer RNA that are found in protein synthesis. mRNA that carry the genetic information from DNA to ribosome where the protein synthesis occurs, whereas the tRNA can transfer amino acids to the ribosome. Catalyzed RNA acts as catalyzed and it is named as riboenzyme. Structural role in ribosome, it is rRNA, and the structural formation of ribosome, it is carried out by rRNA. The regulatory role, siRNA, miRNA, antisense RNA, then processing RNA, like snRNA, processing for the preparation of mRNA and preparation of the rRNA. Then DNA, deoxyribonucleic acid, it is negatively charged, may be single-stranded DNA and double-stranded DNA. The single-stranded DNA is rare in cases, but the double-stranded DNA is common. Negatively charged, it is due to the phosphate group. DNA, it is made up of nitrogenous base, pentose sugar and phosphate group. Due to phosphate group, it is negatively charged in nature. DNA derived from deoxyribonucleic acid. The stable form of nucleic acid due to the lack of 2'-OH group. The pentose sugar here is that the pentose sugar in 2' position, the OH group is absent. So it is named as deoxy at the 2' position. Due to lack of OH group, it is stable in nature. We know that DNA is a genetic material and it is scientifically proved by many scientists. First, the experiment was taken by the Griffith. He take a bacterial strain that causes pneumonia, named as Streptococcus pneumonia or Diplococcus pneumonia. He inject the bacterial strain to a mice and observe whether the mice is alive or dead. He taken two strains, one is R strain, another is S strain. R for the rough strain, a rough surface, and it is encapsulated and not virulent, so the mice is alive. Next, the S strain, the smooth surface encapsulated, it is virulent and cause death. Pneumonia is seen. Then, in the third experiment is taken heat kill S strain. Here the S strain first undergo heat kill. Heat is provided, so the death causing factor is killed. So there is no mice is dead and all the mice are alive. Then R strain. Then he mixed both the R strain and the heat kill S strain, and they found that the mouse was dead. Griffith concluded that all the bacteria that in the heat kill process, but there is some factor is left behind which is responsible for the virulence, and that acquiring by the R strain, something is transferred from heat kill S strain to the R strain that make it virulent. The nature of the transforming factor is DNA. He concluded that the nature of the transforming factor DNA type ही होते. Then the factor is DNA. It is experimentally proved by another three scientists, McLeod, Avery and McCarthy. In his experiment, here is the total experiment. In his experiment, they taken the heat kill S strain bacteria. The bacteria, they contain protein, RNA, DNA, liquids, then sugar, carbohydrate, etc. First, the processing and extract out the liquid and the sugar, that the carbohydrate, and leave behind only the protein, RNA and DNA. So in the further the experiment, only the protein, RNA and the DNA is transferred, and we conclude that anything can be the genetic material. First, in the test tube, they taken proteinase that can destroy all the protein present in it. Only the RNA and DNA is present. Then এই RNA और DNA যেন ଅଛି ଏଟାକେ ଆର୍ଟ୍ସ ଆଣ୍ଡ ବ୍ୟାକ୍ଟେରିଆ ସାଙ୍ଗେ ମିକ୍ସ କରିଦେଲେ ତାପରେ ଇତାକେ ସେମାନେ ବ୍ୟାକ୍ଟେରିଆକେ कल्चर के देन ट्रांसफर करके डेगे से माउस ऊपर इंजेक्ट कले बोले हियर इज द एस्ट एंड एपियर द माउस इज डेड माउसटा डेड है मैंने इन प्रोटीन इन क्लियर है कि प्रोटीन द जेनेटिक मटेरियल नु हिस्से then another test tube रे RNAs मिसाले जेंते कि RNA completely destroy गला then that liquid can mix with the earth and contain test tube that the solution is applied injected to the mice here the S10 is appear सेनो भी से virulence वाला death causing factor appear है mouse मैंने death है बेले इनु भी कंक्लूजन से आसले कि प्रोटीन तो नुहे जेनेटिक मटेरियल ता संगे संगे आरएनए भी नु पहुँचला रेस्ट कैन डीएनए then in the last they can use the DNAs that can damage or destroy all the DNA only the protein and the RNA can transfer and mixed with the yeast and bacteria solution that solution can apply to or inject to the mice and here the nose yeast is appear no transforming factor is there and hence and the three scientists concluded that the nature of the transforming factor is DNA in nature. And the third scientist, Hershey and Chase experiment, they can also prove that DNA was a genetic material that proposed. In his work, they work on the bacteriophage. Bacteriophage is a virus that infects the bacteria. Example, T2 bacteria. Bacteriophage having two components. Only two components. One is protein and another is double-stranded DNA. There is no carbohydrate, no RNA, no lipids, anything. So further, when the experiment proceed, the experiment proceed हवा तो हमें लास्ट कंक्लूजन रे पामा कि आइदर प्रोटीन टा जेनेटिक मटेरियल हेतबो ना तो डबल स्ट्रैंडेड डीएनए टा जेनेटिक मटेरियल हेतबा. First, the experiment. In its first experiment, the protein, this is a bacteriophage. Here it is a genetic material encapsulated by protein coat. The protein coat of the phage, bacteriophage, are radioactively labeled. Here the protein and the sulfur protein used is radio level protein मतलब S35 taken over. Then the bacteriophage is entered into a bacteria through the lytic cycle. Many bacterial progeny are released out. The